Okay, welcome back, everyone. At least I hope you're back. It's my pleasure to kick off this next session. I'd like to welcome Dr. Claire Colas to the ACD and to her first CCDS symposium. Dr. Colas has been focused on studying the structure of solute carrier transporters. Specifically, she uses computational methods such as homology modeling and molecular docking to explore the structure of solute carrier transporters. Dr. Colas will be talking to us today about novel structural models of the creatine transporters and their structural determinants of binding. And Dr. Colas, if you're here, please go ahead and unmute, and you can share your video as well as your screen to start off our next session.
Yes, I'm trying to share. Sorry, this is not what I want to show. Yes, can you see my screen?
Yep.
Okay, perfect. Okay, so hi everyone, thanks for tuning in. Thank you for the introduction. So my name is Claire Colas. I work at the University of Vienna, and I would like really to thank the organizers for giving me the opportunity to present my work today and to invite me because I'm very honored and grateful to participate in such a great event. So my field of research is to structurally characterize SLC transporters in general as a family. And so for this reason, I will start by giving a general introduction on SLCs and explain you why these proteins are emerging and key drug targets, and hopefully convince you that structure-based studies are powerful to target them specifically and efficiently. And then I will go on into more details into our recent work on the creatine transporter to understand its structural determinants of binding with its ligands. So let's go now. So in human, we have more than 500 members of SLCs that are grouped into 65 families, and they are responsible for the transport of many solutes into our cells, such as ions, nutrients, or metabolites. And so they are linked to many diseases and disorders, and for this reason, they are key drug targets where they can be targeted themselves if they are themselves responsible for a disease, or mediate the transport of a drug inside the cell to target an intracellular protein. And in terms of structures, we have a few structures of human transporters available, and this number is increasing thanks to the improvement of cryo-EM methodologies. And we also have access to structures of prokaryotic homologs. And in particular, here you can see a few examples of these prokaryotic structures. And you can see that they can adopt a large variety of folds. And despite this pool of folds, it has been shown that 3 folds are mainly represented. And here you can see an example of those 3 folds. And to each fold, a transport mechanism has been associated. And this is very important to understand because by essence, transporters are dynamic proteins as they need to go from an outward open conformation, as we call it here, to an inward open conformation to capture and release the substrate from outside to inside the cell. And so to conduct structure-based studies, we need to have access to as many conformations as possible of the transport cycle. So here you can see the 3 main transport mechanisms that have been described associated to each specific fold. And as I've been working on SLC transporters for several years now, of different folds, different families, and transporting many different substrates, I've laid out a few key questions to guide my research. So first of all, we want to understand what makes a substrate that is transported inside the cell and what makes an inhibitor that binds to the transporter and prevents the transport of endogenous substrates. And answering this question helps me to understand what makes a substrate with optimized transport properties and how to design inhibitors with higher affinities. Then I would like to discover new ligands for SLC transporters. And these new ligands can be used as chemical tools to understand their function or as new scaffolds for optimization and in the longer run to design new drugs potentially. And finally, as I just mentioned, I would like to understand the transport mechanism, because this could help to understand the structural basis of selectivity of each transporter, and finally to discover conformation-specific modulators. Because a molecule that will bind to an outward open conformation will not be the same as a molecule that would bind to an occluded conformation, for example. And so, as I've mentioned before, I've been working on distinct transporters of different families and folds, and I've tried to answer these questions for each of them. And recently, I've been interested in working with the SLC6 family, which is one of the families that is the most studied due to its high pharmacological impact. And this has been discussed a bit before by Dr. Castor. So the SLC6 family contains 20 transporters that are grouped by regrouped into 4 subgroups. And notably, the monoamine transporter subgroup that you can see here is one of the most studied subgroup of SLCs, as it contains the serotonin, norepinephrine, and dopamine transporters that are linked to addiction and depression. And then we have the GABA subgroup here, to which the creatine transporter belongs to. And then we have the amino acid subgroups. And in terms of structure, these transporters have the so-called Luti fold. And it has the name of the prokaryotic homolog transporter of leucine that contains 12 transmembrane helices that are divided into 2 domains, a static domain shown in pink, light pink here, and a dynamic domain shown in darker pink that opens and closes to capture and release a substrate. And recently, the structures of the human serotonin transporter were solved. And together, these structures provided a lot of information to structurally characterize and study the entire SLC6 family. So now I'm going to go into more details into our work on the creatine transporter. So as I've been saying from the beginning, to conduct structure-based studies, we need to have access to a structure of the protein we are working on. And in this case, we do not, unfortunately, have the structure of the human creatine transporter. So we rely on a method that we call homology modeling, which has for goal, whose aim is to build the structure of the protein we are studying. So in this case, a creatine transporter, using structures of prokaryotic homologs. And so this is a protocol that we are generally using, and I will go through the steps very briefly. So first we need to find the appropriate template, meaning the structure we are going to use to build the model of the target protein. So there are some software that are doing this automatically. And in general, I use HHPRED. And then we align the sequence of the protein, of the 2 proteins, template and target. And here I have to emphasize that it's a very important step, because if the sequence alignment is wrong, then the model, the final model will be wrong. So there's a lot of attention at this point. And then we actually build the models, and the final 2 steps are about optimization and refinement. And these steps are all iterative until we have a model that we can rely on and that we are satisfied with. And so, of course, here I'm not going to go into details, but I'm happy to answer any questions. So for the creatine transporter, I started to build a sequence alignment of the 20 members of the SLC6 family together with the prokaryotic transporter lutein. And interestingly, we saw that in the transmembrane helix number 10, there was a specific insertion that we see here with this serine that seems to be specific to the GABA subgroup of the SLC6 family. You can see that only the transporter of this subgroup have this insertion. And in literature, this insertion has been described previously for the transporter GAT1 and BGT1 as a signature of a pi helix at this position of the transmembrane TM10. So a pi helix, as a reminder, is a turn of helix that is a bit looser than a traditional alpha helix, as it contains 4.5 4.4 amino acids per turn and has a hydrogen bond connectivity a bit longer. And so based on this information and seeing that this seems to be really specific to an entire subgroup, we thought that it would make sense to build a PyHelix at this position of the creatine transporter as well, as it seems to be important to specifically accommodate the packing of the GABA subgroup in this area of the binding site. Then we had to choose a template we were going to use to build a homology model of the creatine transporter. So as I showed previously, we had two choices. So first we could choose a transporter of leucine prokaryotic homolog or the human serotonin transporter. And so when we choose a template, we have to think of various criteria. So one of them is, of course, a sequence identity between the prokaryotic homolog and the target that we are working on. Second, what is important is also the resolution of the crystal structure we use to build the model. And finally, for SLCs, it's very important to also take into account the conformation. And so to start with, we decided to build models on using both templates. And so this is the protocol that we used that is basically the same as the general one I showed, but this one includes a loop modeling, a loop refinement protocol in order to include and build the PyHelix in the specific insertion of the transmembrane helix 10 that I was showing, which is in this area here. So in the end, we obtained two models in two conformations, one occluded conformation here and one outward open. And you can see that the main distance, as expected, is a significant change in the volume of the binding site. So once we had our models, our next step was to try to rationalize the effect of transported substrates using these models. So then for this specific case, we decided to go on with the occluded conformation, as it is more likely to depict the interactions of the model with the transported substrates. So what we did is that you can see here the binding site of the occluded conformation of our homology model. We conducted induced fit docking experiments, which allows the side chains of the amino acid constituting the binding site to move, to accommodate in a better way the ligands that we are docking. And also, based on literature, we decided to deprotonate the cysteine 144 that is part of the binding site here, as it was described in literature to be essential for interacting with the ligands. On the ligand side, for the creatine transporter, it is known that the ligands have two specific moieties, a carboxylate and a guanidine moiety, and that they are linked by a carbon linker that can be 2 to 3 carbon long. And so despite a very similar scaffold, it's interesting to notice that these ligands have a wide variety of transport activities. And so we decided to investigate the influence of the length of the carbon chain here on the transport activities. So we picked a few known ligands, including, of course, the creatine, and 2 potent inhibitors and 2 less potent. And we conducted our docking experiments, and we also predicted binding affinities using MMGBSA methods. And we could see in general that we were able to reproduce the general ranking of the experimental values, only with one exception, the gamma GPA here, which seems, based on our experiments, to be a potent inhibitor, whereas with the IC50 we see that it's not. And I will come back to that in 1 second. So in terms of interactions, we could see a consistency of interactions between our ligand and the binding site. So we could see that the carboxylate moiety interacts consistently with the conserved glycine 73 and with the sodium ion, whereas the guanidine moiety interacts with the cysteine 144 that I mentioned before. And it seems that the 2 carbon lengths between those two moieties is optimal because it allows the perfect interactions with the key residues of this binding site. And it seems that 1 carbon is too short and that the interactions that we just mentioned are weaker, whereas 3 carbon seems too long. And in this case, we can see it actually on our binding pose. We can see that the ligand is very constricted and folded in the binding site, which leads us to conclude that this pose is not reliable, as it happens sometimes in computational studies. And we have to conclude that the scores that we obtain in this case are not reliable as well. And this is the reason why we were not able to reproduce the experimental IC50s observed. So, as a conclusion here, I would like to end by saying that I hope I convinced you that we were able to a little bit further characterize the creatine transporter. We are able to show that it contains this characteristic pi-helix in the transmembrane helix 10, that the cysteine 144 is deprotonated in the binding site. And second, we were able to explain why a 2 carbon linker is an optimal length between the carboxylate and guanidine moieties for optimal transport. And together, the complementarity between these specific features of the ligands and the physicochemical properties of the binding site confers the unique transport properties of the creatine transporter. And so this provides another building blocks towards understanding the SLC6 family altogether, and to understand a little bit better how we are able to observe diversities of interactions and transport within a conserved fold. And to finish, I would like to say that understanding the structural determinants of interaction of the creatine transporters with its ligand paves the way towards a better understanding of its transport process, and that ultimately it will help to understand and rationalize the mutations that lead to transport deficiency. And even though this work doesn't address directly transport deficiencies, I hope that it will be a good basis to discuss and build new hypotheses on how to rescue the effect of such mutations. And I'm looking forward to engage in such discussions with all of you. So you will see the details of this work in the papers that we recently published in scientific reports. And I would like to end by thanking the co-authors of this paper, as well as the pharmacoinformatics group of the University of Vienna. And I thank you all for your attention.
Thanks so much, Claire. That was a lovely talk. I'm sure we'll have questions for you. We will get to questions after the next talk. And so here, I'm just going to bring up a brief introduction for our next speaker. So our next speaker is Dr. Emil Alexov. He is